Hello everyone. Today we are going to dive into the topic of protein degradation, a crucial process in cell biology that helps maintain cellular homeostasis. This presentation will cover the various pathways involved in protein degradation and how cells use these processes to regulate their internal environment, respond to stress, and ensure proper functioning. So, protein degradation serves multiple purposes. It's a way to digest material brought in by endocytosis. It helps discard unnecessary proteins, such as those from synthesis and folding mishaps. It slows or stops biochemical reactions when needed. It plays a role in regulating metabolism through processes like autophagy. And finally, it's involved in presenting internalized antigens within immunological cells. So there are basically two types of protein degradation. One is lysosomal, which of course we know is a cell organelle. So it's an acidic organelle. And then another type of protein degradation that doesn't occur in lysosome, rather it occurs in the cytosol. So various enzymes are involved in that case. Now, degradation in lysosome. Let's start with heterophagy, which is the lysosomal digestion of extracellular material. When cells take in external substances, they form endosomes or phagosomes. These structures then fuse with lysosomes to create phagolysosomes, where the engulfed material is digested. Next, we have autophagy, a process that maintains cellular balance by degrading proteins and recycling damaged cell organelles. Autophagy can be particularly heightened during cellular stress. So here you can see the degradation pathways in lysosome. There are in my case of macroautophagy, there are pre-autophagosome. In case of microautophagy, there are lysosome. There is also cytosolic chaperone complex, ubiquitin, proteasomal degradation, and the normal endocytosis that only involves the plasma membrane. So the five different mechanisms are endocytosis, clinophagy, macroautophagy, microautophagy, direct transmission or translocation from cytosol. Now, there are three main types of autophagy: macro, micro, and chaperone mediated. We will first talk about macroautophagy. It's the main pathway where a double membrane forms around damaged organelles and proteins, and it's a more complex and regulated pathway. In case of microautophagy, it involves the direct engulfment of cytoplasmic material by lysosome. Unlike macroautophagy, it doesn't involve autophagosome formation. This pathway typically targets proteins containing the KFERQ motif, which is basically lysine, phenylalanine, glutamic acid, arginine, and glutamine. The last type, CMA or chaperone mediated autophagy. It is highly selective and complex. Here, proteins must have a specific recognition site to bind to chaperones. This substrate and chaperone complex moves to the lysosome membrane when the protein is recognized, unfolded, and then translocated across the membrane for degradation. There's also a lesser known process called clinophagy, where secretory granules are directly degraded and recycled without being released. Now, let's break down the steps of macroautophagy. Okay, so first, induction. The process is triggered in response to cellular needs or stress. Step number two, vesicle nucleation, also known as phagophore formation, where a vesicle starts to form and elongate. Step number three is vesicle elongation and cargo capture. So, what happens basically, the vesicle selectively or randomly captures targets for degradation. Step number four is fusion with the lysosome. The autophagosome merges with the lysosome, where enzymes like uh, cathepsin proteases B and D break down the contents. Lysosomal membrane proteins LAMP1, LAMP2 play key roles here. So these proteins, cathepsin and LAMP1, LAMP2, these proteins sometimes come in entrance examination. So these steps are really, really important. Okay, so next part is cytosol degradation. Uh, so now, when proteins are synthesized, they begin folding immediately. Chaperones like HSP70 assist early in the folding process, while HSP60 ensure quality control once folding is complete. So here you can see the HSP70 machinery and also HSP60 machinery. HSP60 works like a quality control chamber after a protein is completely folded, and HSP70 works to help fold early in the life cycle of a protein. Now, as I've already said, in the cytosolic degradation, there are many enzymes involved, and the most important protein involved in cytosolic degradation is ubiquitin proteasome pathway. So, proteasomes are a major mechanism by which cells regulate protein concentration and degrade misfolded protein. They contain a proteolytic that handles degradation, and while there are caps that regulate ATP hydrolysis and entry of substrates. So, additionally, cells can regulate protein degradation by activating ubiquitin ligases, which tag proteins for destruction. Generally, proteins hide their degradation signals, but there are various mechanisms that can expose these signals when necessary. So, this is the structure of proteasome, which is a major mechanism by which cells regulate the concentration of particular proteins and degrade misfolded proteins. So, in summary, protein degradation is a complex and highly regulated system essential for cellular health. Whether through autophagy, proteasomes, or specific chaperones, these processes ensure the proteins are properly managed, allowing cells to adapt and thrive in different conditions. Well, thank you for watching, and I hope this explanation helped clarify the fascinating world of protein degradation. Thank you.